This Week in Science and Education is presented in association with the Science Coordinators and Consultants Association of Ontario. Visit their website at sccao.ca.
This Week in Science and Education is brought to you in part by the University of Western Ontario, www.uwo.ca. We thank them for their support. This Week in Science and Education is also brought to you by Laurentian University. Check out Laurentian at laurentian.ca. We thank them for their support.
Hi everybody, welcome to this week of TWISE, episode 73. I'm here with Dr. Kang, who's going to tell us a little bit about his research in HIV. So Dr. Kang, why don't you just jump right into it for us?
Thank you. Good afternoon, everybody. I am a virologist here at the University of Western Ontario, and we developed a vaccine against HIV and AIDS. As of today, there are about 34 million people living with HIV infection, and we have lost about 28 million people over the 2.5 decades due to the HIV infection. And people have been trying to develop vaccine against this HIV, and there have been 3 trials in the past, but unfortunately, none of those 3 vaccine trials have been successful. And we are developing vaccine using the different strategy, but which has had the proven record of success with the other viral diseases, such as polio, hepatitis A, flu, rabies, and so on. So we developed vaccine using the strategy known as a kill the whole virus strategy. So what we did is we genetically modified the virus gene. This, what you can see, can you see it?
So you just need to...
This one here.
Yeah,
Make it full screen.
And
there.
now this is the structure of the genes of HIV. And now this one has 3 major structural genes, GAG, POL, and M. These are the components of the virus. And then there are other non-structural regulatory genes. What we did is we deleted NEF gene, the red one, and this gene is responsible to produce disease after infection of the HIV. And then the green one produces the surface glycoprotein, that's the surface antigen of the virus. And to synthesize this green one, you need a signal peptide to govern that the synthesis and intracellular trafficking. So what we did is we deleted this gene, NEF gene, responsible for pathogenesis, and then in order to enhance the production of HIV much more efficiently, we replaced the signal peptide of the glycoprotein, that is a green protein here. And then as a result of the replacement of that signal peptide, we are losing the VPU gene. And that's the VPU protein overlaps with the signal peptide. So we are losing VPU gene. So this virus is now NEF negative, VPU negative, and signal peptide gene replaced. So we have produced this virus in large quantities by infecting the cultured lymphocytes. And those infected cultured lymphocytes can produce large quantities of virus. And we purify those and inactivate the virus by using 2 different methods. One is a chemical method, that is 2,2-dithiodipyridine. This chemical cross-links internal proteins of HIV. And then to make sure that it is completely killed, we are using the gamma irradiation to irradiate the virus so that we can kill the virus. So we're using this kill the whole virus as antigen to produce the immune responses in vaccinated individuals. And we have gone through very elaborate tests of this vaccine and to make sure that it is safe. So I think I didn't have that slide. And so that we did all the tests, and the test involves about 230 different tests of this vaccine to make sure that it is safe putting into the human. And US FDA has now approved this vaccine to be tried in human. And so as of the December of 2011, we have now approval from US FDA for the human clinical trials. And we are now starting the phase 1 human clinical trials in the United States. So that's what we have today.
Wow, that's really great. Dr. Kang, I know that we've been trying to make a vaccine for, over 30 years? And we've hit roadblock after roadblock. Can you talk a little bit about why it's been so difficult to create a vaccine in the first place, and what it is that is so different with your approach that has allowed this to work and get this far along in the process? So 2 parts to the question: why has it been so difficult in the first place, and what is it that's so different about the approach you've taken that has allowed you to be successful?
Okay, the question number 1, why is it so difficult to produce the vaccine against HIV? No one seems to know what antigen or what component of the virus that one should use to create the vaccine against HIV and AIDS, which can prevent HIV infection. And so selection of antigen is the problem. Second, there is no animal model to test the efficacy of the vaccines if you have experimental vaccine to test it, because none of the animals, including non-human primates, to get infection with HIV, so that we cannot test the efficacy of these vaccines. And thirdly, that there are 3 different types of the vaccines that one can produce. One is live attenuated virus vaccine. That cannot be used for HIV because HIV replication goes through so-called proviral DNA stage, and that proviral DNA integrates into the chromosomal DNA and establishes persistent infection. Therefore, it is not desirable to have a viral genome integrated into the chromosomal DNA. So live attenuated virus vaccine choice is out of the question. And that has been tried in the monkey AIDS virus, known as SIV, simian immunodeficiency virus, and that works, but not for the human. It is not possible to use a live attenuated virus for HIV vaccine for human because we can activate those silent genes, such as cancer genes, when you integrate the chromosomal DNA, the live virus genome into the chromosomal DNA. So therefore, we cannot use live virus vaccine strategy. The second strategy is a kill the whole virus strategy. That has been successfully applied for the production of the polio virus, the flu virus, hepatitis A, rabies virus, and Japanese encephalitis, and so on, and also some 16 different animal vaccines using the kill the whole virus strategy. But no one has tried this strategy for the HIV vaccine development until we have done it. And the third group of vaccine is a subunit vaccine, such as hepatitis B virus vaccine is based on surface antigen, which is 22 nanometer particles of the hepatitis B virus, and also the human papillomavirus, which is based on L1 protein of human papillomavirus, which is major structural protein. And those subunit vaccines work. But when the vaccine has tried the surface antigen of GP120 as a vaccine, it did not produce neutralizing antibodies. So that failed. And then now the newly developed vaccine strategy is a recombinant virus vaccine strategy. In other words, we can get the virus which can infect humans, but it doesn't cause any disease in humans, such as human adenovirus 5. The Merck has tried to use human adenovirus 5 and then putting the gene of HIV into human adenovirus 5 and tried to vaccinate people. It turns out that those people who had pre-existing antibodies against the human adenovirus 5, they had a higher rate of infection than those control groups. So they had to stop it in 2007. The other strategy that is used for recombinant virus vaccine is using the animal vaccine, animal virus, such as Canary pox. This is pox virus carrying the HIV gene, and that was tried by Sanofi Pasteur in Thailand, and they used about 80 to 100 people in each arm to vaccinate people the differences was only about 30%. So that was not effective.
Yeah.
So we are using the strategy, which is kill the whole virus strategy, and for the vaccine development. That has a proven record for the other viral diseases. One may say that, oh, retrovirus is different from other viruses. Of course. But no one has tried this. And certainly our body, when it receives a foreign substance, such as a virus vaccines, it will react to make humoral immune responses. So this vaccine will certainly make the good antibody response, but it may not produce much of the cytotoxic T-cell response, which is known as a cellular immune response. But the humoral immune response is good enough to protect people from infections. And that has been proven in so many different cases.
Okay, that actually last bit was started to answer one of the questions that I had, and it had to do with how HIV being a disease of the immune system and how it targets the very cells that are helping fight disease. And so just maybe get into that a little bit in terms of, so if someone gets the vaccine and starts to produce the antibodies, sort of what will be the success rate that it will be able to fight off an invading live virus before it replicates enough that it sort of then gets to the point where it can't fight it back off again? Like, are you trying to do like a complete eradication, or is this going to be sort of a suppression in the case of infection? Can you talk a bit about that? I'm not really sure I understand it.
Well, there are two different types of the vaccines you can develop. One is for the prophylactic vaccine, that is preventive vaccine, and kill the whole virus strategy is to prevent infection, so that if you immunize naive people, those who are not infected as yet, and you can trigger immune responses, and they will produce antibodies against all those viral antigens in the structural proteins, including the surface antigen. And if you make antibody against the surface antigen, that will neutralize virus infectivity. Therefore, you can prevent the virus infection when it comes with the same virus. And now that is different from the therapeutic vaccine. And therapeutic vaccine, that we are trying to induce a cytotoxic T-cell response. That is, the CTLs can recognize, those are the CD8 positive CTLs can recognize virus-infected cells. And virus-infected cells will have the histocompatibility antigen is called MHC class 1, which will combine with a small peptide of the virus and make a MHC viral peptide complex, which will be expressed on the surface of the virus-infected cells. That can be recognized by CD8 positive cells, which have been isolated from the vaccinated people, and that can recognize those infected cells and destroy it. And that's the way we can clear the virus infection. So here we are talking about the two different strategies, the strains of the virus, and one is for the preventive vaccine. We are mainly targeting for the humoral immune responses, and then we have also developed the vaccine, which now is being tested in animals. That is for the treatment vaccine, is the therapeutic vaccines.
Wow. Okay.
Excellent.
Thank you.
Before anyone asks another question, we're going to stop for a break. So we'll just get a word from our sponsors, and we'll be right back with Dr. Kang.
This Week in Science and Education is brought to you by Sheridan Institute of Technology and Advanced Learning. At Sheridan, students shine brighter. Check out sheridanc.on.ca.
So welcome, everybody. We're back here with Dr. Kang, and he's just telling us about his research for the HIV vaccine. So first, I just have to say congratulations. You've made monumental steps, and this is absolutely wonderful.
Big news.
So maybe tell us a little bit before we get back into the research what the next steps entail. How long before patients are actually able to get this treatment?
Okay. It may take about 5 years. The first year, we are going to use you're going to use the HIV positive healthy individuals to test our vaccine for safety. And that's what's known as a phase 1 human clinical trials. And you don't need many people. It's about 40 people that we can recruit. Those are the HIV positive healthy individuals. And we'll test this vaccine in those people and see if that vaccine doesn't cause any adverse effects. And then once we have done that, then we can go into the phase 2 human clinical trial. And phase 2 human clinical trial is looking for the immune responses. That may take about another year. We are going to recruit about 600 people, 300 people in each arm, and we'll look at the immune responses. We know up to that point we have no problem because we have done this in non-human primates, and this vaccine actually induces some real good antibodies. Excellent. Now the phase 3 human clinical trial is really the efficacy test. See whether or not this vaccine can prevent HIV infection. And that's the efficacy test, and that takes 3 years' time because you immunize people and you have to wait for the natural infection. You cannot challenge them with the live viruses, right? That's unethical. So we have to immunize people and wait for 3 years and see what's the difference between the control group and vaccinated group. And if the difference is remarkable difference, then you have efficacy. So that then we can really release this vaccine for actual use to prevent HIV infections.
So I have a question following up on that. So in this, the phase 3, you're faced with a really interesting ethical dilemma. If this vaccine works, you're going to be vaccinating half the people that are at risk, and the other half are not vaccinated. So how do you handle the ethics of that kind? I mean, we don't have any choice because you've got to test to see whether it works. But how do you handle the ethics of this kind of step where you've got a treatment and you can't treat everybody?
Unfortunately, those people who belong to the control group will lose their chance, right? And so that people will be immunized with the saline alone in a control group, and those people who will be vaccinated with the real vaccine. And this is what's happening in any kind of human clinical trials. You need the both arms. One is control group and the other one is the actual vaccine group. So it has been done, all trials, the human clinical trials, in not only vaccines, but also all kind of medications that we have.
Absolutely.
So I have a question. Oh, go ahead.
Let me just follow up quick, just because it's the second half of that question. Is there any indication that people behave differently once they're part of the trial? So they're more reckless because they think they may be vaccinated?
That was my question.
Because it's not going to. Well, we are not going to disclose who are in the control group and who are in the vaccine group.
Absolutely, but you think if I've gotten the vaccination, I think there's a 50-50 chance I've been vaccinated.
That's correct. But unfortunately, we cannot disclose that because this has to be double-blind, the control, double-blind.
I completely understand that. What I'm asking is there any indication that people do behave differently once they're part of this study, where they think that, okay, there's a 50-50 chance? I understand that it has to be a double blind, but what do we know about people's behavior as part of these studies?
All the volunteers for any kind of human trials, they sign up for the no complaints, right? And in fact, they do accept the possibility of being in a control group and possibility of being in a vaccine group. So once they sign that consent form, and in fact, that's how the clinical trials are conducted.
Yeah.
So, I see what you mean. Yeah. But that's the way it has to happen, because it has to be double-blind, placebo-controlled trials.
So is there a chance that 600 people go out and none of them are exposed to HIV?
It is possible, but no, 600 people is for the phase 2 human clinical trials. And for the phase 3 human clinical trials, we are going to recruit the 6,000 people.
Oh, 6,000, excuse me.
So that the 6,000 people will have 3,000 in each arm.
Right.
Okay. And so they don't know whether they get the real vaccine or just a saline.
Right.
Right. Until we decode it.
But once they move past that stage, is it possible that all 6,000 people, how do you track that? Like all 6,000 people could go out and potentially none of them could be exposed to HIV. Maybe the opposite of what Thomas is talking about happens and they're all thinking more about HIV and they're all...
No, the recruitment, that there is a certain criteria for inclusions, right? The inclusions that you have to belong to one of those 4 groups that we call high-risk group. Okay. These include hemophiliacs, and they need multiple blood transfusions, so they have a better chance to get infected, right? Although our blood supply system is very safe, but you never know. So those people who get the multiple blood transfusion are classified as a high-risk group.
That's interesting.
Okay, and then those people that were using the injection drug abusers or injection drug users, and they are classified as a high-risk group. And the female, especially female sex workers,
right,
that they classified as a high-risk group. And finally, the homosexual gay people with multiple sexual partners, and they classified as a high-risk group. So those are the 4 groups of people that we are going to recruit. And the recruit, actually the inclusion criteria has to be very specified, and then everybody will actually meet that requirements.
Right. Okay, that makes more sense to me.
So that if you are recruiting only just the ordinary people, you are right that they may never have a chance to get the infection, right?
That's right.
And therefore, you have to recruit those people who has chance to get infected.
Right. Colin, you've been pretty quiet. Do you have any questions?
Yeah, I'm just when you get to the well, I guess in all phases, where exactly are the phases happening? Is it you said you got US FDA approval? Is it happening in the States somewhere that these trials are occurring?
Yes, the phase 1 human clinical trial is being arranged in 2 cities in the US, and the phase 2 and phase 3 will be multinational.
Oh, okay. Interesting.
So I've got one last scientific question. I think we're probably running out of time, which we almost always do. You mentioned there's no animal model for this system. What about cell lines? Is there a reason that has work been done on cell lines? Can you do work on cell lines?
Well, we can produce viruses in actually cultured human T lymphocytes. There are many different lymphocyte cultures there that we can infect with HIV, and they produce viruses. But you cannot just the cell lines do not produce antibodies. Okay, so
of course
we are looking at productions, so that we are looking at the live animal model, and there is no live animal model, so therefore we have to use human model or human trials to look at the protective immunities.
That makes a lot of sense. Thank you.
Yeah.
Excellent.
I'm gonna ask one education question, if I can, to wrap up, Nicole.
Yeah.
I deal a lot with secondary science students and high school teachers and that sort of thing. I'm just wondering, from your point of view, if you were to, talk to, say, a secondary biology class about the importance of this work and how you go about this work, what would you sort of distill it down into? Just a few moments of, this is the core of what we do, and a simple explanation to some high school students.
Well, I can tell high school students that we are using not the unknown technology. We are using proven technology to produce vaccines, and this technology has been used over the years, decades, to produce vaccines for very important human diseases such as polio, flu, rabies, hepatitis A, and so on. And so we are using the same strategy, but in order to make this vaccine, we had to modify genetically so that we can produce large quantities of less dangerous virus. So that's what we did. Suppose we are successful in this strategy, then we can save millions of lives with these vaccines around the world. And as I mentioned earlier in my intro, that there are about 34 million people living with HIV infection. And if you can really save those lives, that would be tremendous achievement. Now, just one example that we have eradicated one very important infectious disease such as smallpox. And when Edward Jenner introduced the smallpox vaccine using the vaccinia virus, that's a cowpox virus, to immunize people to prevent smallpox infection, and he saved millions of lives over the years. Now there's not a single case of smallpox on the surface of the earth today. As of 1978, it is completely eradicated. Now that is the biggest achievement in infectious diseases in human life.
Yeah, and the potential of your work is amazing. I mean, I was, grew up in the post-AIDS era. I went to high school in the 80s. This has been a part of my life as long as I've been an adult. And the idea that we don't have a vaccine has been part of my life the entire time I've been an adult. It is really exciting to know that we're getting closer. And I really appreciate you taking the time to explain this to us. It's some neat science that allows you to do this work.
No, it's my pleasure, Thomas.
Thank you so much, Dr. Kang.
Thanks
It's my pleasure.
Thank you. For This Week in Science and Education, thanks for watching, and we'll see you next week.